Hello, Biology 11. So hopefully as per the email, you've started by watching those first two videos. And if you haven't, let me just point that out again here, 1 second. So as I said in the email, just go to our course Moodle, go to the book of videos for chapter 12 on microorganisms, and I'd like you to watch video number 4 and 5 on what is a virus and viruses. Those will also be helpful for your project, your case study that you're working on the pandemic pandemonium. But for the notes, I'm just going to kind of go through these briefly, and I'll also get you to do a few things. So looking at a typical virus, they are very simple in nature and design. Um, and some interesting tidbits about viruses that I don't know. I've heard lots of different numbers in terms of the total number of different kinds of species of viruses, in the hundreds of thousands. But there's only about a little more than 200 different viruses that infect us, which is still quite a bit. But you also have to think that just the common cold and the number of different rhinoviruses make up a substantial number of that, right? But there are a lot. Some other weird, interesting tidbits. If you remember last time when I talked about how small viruses are, so remember our cells, eukaryotic cells, largest cells that exist, and then you have bacteria, which are probably 1,000 times smaller, and then you have viruses, which are 1,000 times smaller than that. But if you took all the viruses within the oceans, and that's another funny tidbit, used to be that biologists didn't believe viruses existed in aquatic environments until somebody, I don't know when it was, maybe 8, 9, 10 years ago, started doing some research and looking at the fact that there are viruses in our oceans. And if you took all the viruses that were in our oceans, as small as they are, and stretched them end to end, tip to tip, they would travel about 10 million light years, which is a number can't even fathom, right? But this number down here, 10 to the power of 31, would give you numbers that we can't even comprehend. Long and short of it, a virus has basically three main components to it. So number one, it has this recognition spike. And this is basically like a protein, right? And we have these on the surface of our cells as well. It's partially what allows a virus to attach to our cells. But it is a protein molecule, and often referred to as an antigen. And antigens are things that can stimulate an immune response, right? But they're also basically recognition markers. I think, if I remember, this was a picture I got offline that is the SARS-CoV-2 virus, right? And this one here, I'm not sure, I can't remember where I got that one from, but basically you've got this protein spike on the surface, and then the capsid represents another protein, generally a protein coat, but it can be made of protein, lipid molecules that represent kind of like the equivalent of the cell membrane on a bacteria or on our cells, but it's not made of the same structure at all. So this structure here represents this capsid, this protective covering, right? And within each virus is basically some nucleic acid. Now remember, nucleic acids consist of both DNA and RNA. So one thing that's really important, this is a bit of how viruses are classified, is there are DNA viruses. So that means that their nucleic acid, their hereditary molecule is DNA. So there's DNA viruses, and there are RNA viruses. And it's based on what type of nucleic acid they house. And then you've even got these retroviruses in which their RNA can be converted into DNA and incorporated into the host's DNA. So lots of weird, crazy characteristics or differences between viruses. All right. The main thing we're going to focus on really is just the way in which viruses reproduce. And this is part of the process or part of the understanding of viruses as being non-living, right? There's viruses have lots of living characteristics in the sense that um, they do contain genetic material, right? These nucleic acids, but the fact that viruses lack the ability to reproduce on their own uh, kind of categorizes them as non-living, right? But they do reproduce in two different ways, or kind of connected together. So although they can't reproduce on their own, right? Viruses cannot reproduce on their own. They basically require a host cell to facilitate reproduction. All right. And basically, the way in which it works is the two different cycles are referred to as the lytic cycle. Okay. The lytic cycle versus the lysogenic cycle. Lysogenic. All right. And they basically differ in terms of the rate at which the virus can replicate inside of a host cell. So this example, or the two reproductive cycles, are shown here, where this guy is your common bacteriophage. So this is the virus, and in this case, it's infecting a bacterial cell. So lots of bacteria viruses are referred to as a bacteriophage. And phage just kind of means eater, right? So this is where set up here, bacteriophages. So what's happening is this. Basically, you get this virus. Let me write the steps down here. So in the lytic cycle, you get number one, attachment. All right. And this is essentially the phage attaching onto the surface. And that's kind of where these recognition spikes come in, these antigens. They can allow viruses to attach to the host cell. Then you get insertion. In many cases, the virus doesn't go inside the cell at all, but rather just inserts its genetic information, inserts its nucleic acid. Now remember that nucleic acid could be DNA or RNA, but it's inserting that nucleic acid into the host cell. Okay, and then number three, you get replication. And so essentially, that nucleic acid, whether it's DNA or RNA, basically is the information to make more viruses, right? And so what happens is the host cell machinery reads the genetic info and manufactures virus parts. Manufactures virus parts or components, which then self-assemble and form more viruses inside the host cell, and that leads to lysis, which, if you remember, this is the word for cell splitting, or just splitting, but in this case, cytolysis, which would be the splitting of the cell. The cell fills with so many viruses that it literally ruptures, releasing those viruses into the surrounding to go on and infect other hosts. All right. So that's the lytic cycle. It can be very rapid. When you think about from time of infection to when you start feeling symptoms, it's already spread to multiple cells, hence your symptomatic response, right? Now, in the case of the lysogenic cycle, which is happening down here, we have the same sort of steps happening. We get attachment, and then we get integration, and it doesn't show it here, but what's happening is the nucleic acid from the virus is being incorporated into the host. And what happens is it then just enters into a state of dormancy. And the host cell doesn't actually manufacture more viruses. It's just taken up the nucleic acid, DNA or RNA, into the host's DNA. And then, of course, that host can undergo, cell division. And all along, when it's dividing, it's got the viral DNA. So it goes into the state of dormancy containing viral DNA, right? Or viral DNA or RNA. But the host itself would have or recognize as though nothing's going on, and it's just sitting there dormant. But at some point in time, some environmental factors will cause it to then switch into the lytic cycle. And all of a sudden now the host cell starts reading the viral DNA or RNA and starts manufacturing more viruses. And then it's hitting the lytic cycle and it's back into rupturing and releasing those viruses on. So it's kind of like this switch between the two. Some viruses infect and they instantly go into the lytic cycle. Other viruses may infect and they just go dormant in the host, but at some point in time, they they enter into that lytic cycle as well and then cause infection. A good example of this is if you recall ever getting chickenpox, which maybe your generation, you guys in Costa probably haven't, but when I was a kid, they didn't have the vaccine for chickenpox. So every parent wanted their kid to get chickenpox. It's far less severe getting chickenpox as a child. And it's a very, very contagious disease. So, very easy to get it. If one kid in the family gets it, everybody's probably going to get it. Or one kid in the neighborhood gets it, chances are everybody's going to get it. But chickenpox instantly enters into the lytic cycle. But what's really interesting about it is some cells may enter into the dormant stage. So some host cells that have been infected by the chickenpox virus enter into this dormant stage, and it could be years, typically 10, 20, 30 years into your adult life, where all of a sudden the chickenpox virus manifests itself into the lytic cycle, and this is referred to as shingles. And so again, it shows the diversity of viruses, how some even can transfer back and forth between this lytic and lysogenic cycle. So that's really all there is to viruses. We're not going to go into a ton, but what I do want you to do is check out in your textbook. So this is your hard copy textbook, the core, and just read about other classes of pathogens. So at this point in our microbiology study, we've looked at bacteria, and of course, all the good things about bacteria, but also recognizing that bacteria are pathogens. Well, viruses are pathogens as well, right? These are things that cause disease. Bacteria and viruses are probably the things you think of most commonly when you think about pathogens and these germs and microorganisms. But I want you to read in your textbook about these other things called prions and viroids. These are essentially animal pathogens that aren't really cells themselves, but they are cell components. They could be sort of protein-like in nature, but they're really not living at all, not like a bacteria cell or anything. They're just, I guess one way to think about it might be to think of, although a pollen grain actually consists of well, a good example would be, say, having a peanut allergy. A peanut allergy induces this immune response because of the nature of the proteins found in this peanut, in this nut, right? It's not a living cell itself. Well, prions are animal pathogens, cell components that can cause disease. All right. And viroids are plant pathogens in the same ballpark, in the respect that they're not living cells, but they're cell components that can cause disease. There's a video on the Moodle, which I'd like you to watch and also read in your text about these, and we can talk about them. You might have heard of some of them. Maybe you've heard of way back in not too many decades ago, the mad cow disease. So mad cow disease is a result of exactly one of these prions that has caused infection. So that's it. Just a couple things. One more thing I would like you to do. You should be working on your case study and plugging away at part 2 in that. And then the other thing I would like you to do is, if you go on to Moodle and you go to the next chapter, which we're going to be starting probably tomorrow, and you guys are going to be doing some of this work on your own. If you go to student notes here and you look at this down at the bottom, a Word doc called Protista Notes. If you click on that, this is a blank doc that you guys are going to start filling in. Don't worry about this top part. I'm going to do it for you, but we're going to start looking at all the different um, representatives of kingdom Protista, and they're divided into plant-like and animal-like. And so you're going to be looking at each phylum. You're going to give me a drawing, and then you're going to describe these characteristics about each one. So you'll be able to do this through the internet, you're using your textbooks, etc. So starting with phylum Euglena you can search a Euglena, you can make a drawing in this space, labeling all of these things here and making sure you describe what they do. And then describe the locomotion. How does this thing move around? Describe some ecological, environmental roles that this organism might play. And then if you come across anything really cool, interesting, then make sure you add that in here. And then the next phylum, Basileophyta, the representative here being diatoms. So again, do a search, do a drawing of a diatom. And in this case, not much to label, just emphasize the nature of the cell wall in diatoms. And again, go through and describe locomotion. How do they move around? Can they move around? What's their environment? What's their ecology? Any interesting facts? So if you can maybe get 1 or 2 of those done, you'd be on a good pace to be getting it done by the end of the week. All right, see you next class.